Breaking Down Biology blog Q&A session. My name is Neha Sharma, and today I am joined by UCSF scientist Dr. Atesh Worthington, the first author of the preprint In Utero Lipid Nanoparticle Delivery Achieves Robust Editing in Hematopoietic Stem Cells. This study explores the beginnings of a potential treatment for hemoglobinopathies in rodents. It's great to have you here today.
Thank you for having me.
All right, so my first question to you is, can you give us some background on what early onset hemoglobinopathies are and what are their current treatments?
Definitely. So hemoglobinopathies are disorders that affect our ability to make hemoglobin, which is the protein that carries oxygen throughout our bodies and ensures that cells are able to be fully oxygenated and perform all the tasks that they need to. There are multiple types of hemoglobinopathies because hemoglobin is a protein that's made up of two different types of globin protein. So there are alpha globin hemoglobinopathies and beta globin hemoglobinopathies. The different ways in which we treat hemoglobinopathies depend on the different type. Right now, for sickle cell anemia, there is an FDA-approved genome editing therapy called Casgevy, where you can take blood stem cells and edit them to fix the sickle cell mutation. For beta thalassemia, this method works as well, but there's also bone marrow transplants that can be used to cure people with different hemoglobinopathies, essentially replacing the blood stem cells that make all of the blood in the body. One of the major limitations with bone marrow transplants is that there are a lot of comorbidities with those types of therapies because you have to remove all of the blood stem cells in the person's bone marrow, either giving some version of chemotherapy or what we call myeloablative conditioning to essentially make space for the healthy donor cells to be put back in. As you can imagine, that's not a good time for anyone. For a lot of bone marrow transplants that are allogeneic, where you're getting a donor to donate their bone marrow to you, it's difficult to find someone that is perfectly matched. There's now a lot of interest in trying to correct cells that are already within the patient's body, so in vivo genome editing.
I thought it was interesting that you were talking about bone marrow transplants because I was thinking that that's probably a really invasive procedure. I was wondering, do you think your approach in your study was less invasive? And do you think you would be able to describe your procedure and why you chose LNPs?
Yes, most definitely. So the goal of using in vivo genome editing is to have a therapy that is significantly less invasive, that doesn't require the harvesting of bone marrow from either a donor or the sick patient, and then the retransplantation of those bone marrow cells. So in the same way that you get your blood drawn when you go to the doctor, delivering a therapeutic that way. Typically in us and in adults, the blood stem cells reside in the bone marrow. That is the niche, we call it, and they're very difficult to access. Typically, you go into the hip or the iliac crest to harvest bone marrow, and it's painful for the donor. Or you can mobilize the bone marrow, so essentially give a patient drugs to encourage those blood stem cells to leave the bone marrow and go into circulation, which is less invasive, but still can take a toll on the donor. What we're interested in doing is taking advantage of development when the blood stem cells are actually in the fetal liver. So those blood stem cells that will eventually go to your bone marrow, the hematopoietic stem cells, as we call them, or HSCs, found in the liver in a mouse and in a human during development, where they expand significantly before they migrate. The idea is the liver is significantly easier to get to with an intravenous injection. And then in the rodents, at least during postnatal life, they're in circulation as well, similar to humans where, when you collect cord blood from neonate, when the baby is born, that cord blood contains the blood stem cells as well. We were injecting lipid nanoparticles that contain different types of genome editors into the fetal liver of different types of reporter mice. And we also complemented that with neonatal injections. So once the fetus was born, at day 1 of life, we could also inject intravenously with our lipid nanoparticles. And one of the reasons we're interested in using lipid nanoparticles is there's been a lot of safety studies with the COVID vaccine that use lipid nanoparticles to deliver mRNA cargoes. So we know that there's quite a bit of safety there. Additionally, we're interested in a technology that has the possibility to redose patients.
Why do you think only 15 to 30% of the HSCs retained edits in utero?
Yeah, so HSCs have just been notoriously difficult to edit. I think what we're up against when we're trying to reach good levels of editing is getting the lipid nanoparticle to actually interact with the HSC and getting enough HSCs to see the lipid nanoparticle. So, and also giving nanoparticle at a dose that is safe and effective. We have to balance safety and toxicity with actual editing levels. If we were able to give the mice a higher dose that was well tolerated, we probably could have reached higher levels of editing in the HSC. But what we were really excited about was that we had some animals in which their levels of editing were at a level that could be considered curative for patients that have different hemoglobinopathies. So right now in the clinic, we think between 20 and 25% editing would be sufficient to treat or to cure these different red blood cell diseases. And one of our mice, I think we got upwards of like 36% editing. But one thing I do want to mention is that once the cell glows red, indicating that it received an edit, that was actually underestimating the true levels of editing, the way the reporter mouse works. So in some of those experiments, we actually likely got significantly higher levels of editing. We think it undercalls levels of editing by threefold, just based on the genetics of how the mouse model works.
Those sound like really promising results. I was wondering, if you were to look back on your study, are there any methods you wish you had approached differently? Or did it spark any new ideas for what you'd like to research next?
That is a great question. What are the limitations and how can we improve on them? I think one of the main things that we are definitely interested in changing when we do future experiments are the ways in which we perform these injections. Something you might notice is the variability in the editing levels when you compare delivering the genome editors either in utero or postnatally. Part of that has to do with the way in which we do these injections. So in utero, when you perform the intrahepatic injections, the surgeons in our lab do their very best to replicate the exact injection each time. But as you can imagine, you see the liver and you put a very, very tiny needle in it, and it's hard to say whether or not it actually made it into the liver. So there's different types of injection methods called a vitelline vein injection, where you directly put the needle into this vein that accesses the fetal circulation. And we're starting to learn how to do those injections with a lot more precision and accuracy, because when we do these injections postnatally, it is specifically an intravenous injection. So we see much less variability in the editing levels of those mice. The other things that we're really excited about are using these lipid nanoparticles actually in a disease setting. Right now, all of the data in the paper is a lot of proof of concept to show that we can actually target HSCs. But in the future, we're really fascinated by and encouraged to try to find genome editing strategies for another type of hemoglobinopathy that I haven't talked about called alpha thalassemia major. We have genome editing strategies for beta thal and sickle cell, but there is nothing yet for alpha thal. So that is kind of the next frontier, actually meshing delivery and a therapeutic edit.
That all sounds really interesting. To our audience, if you are interested in reading more about the study, please visit our bio. And thank you so much for joining me today. I really appreciate it.
Thank you for having me.